This is our first of 3 micro lectures for chapter 12. And this one's going to be focusing on nervous tissue. In this lecture, we're going to be looking at what is a neuron and how does the neuron use myelin. So let's get started. The nervous system is one of great complexity. It's very, very, very complex because it has many different features. So you need to have the different types of cells within it to make sure that we can sense heat, pain. We can affect muscles. We can affect glands. And by doing this, the nervous system is now the foundation of our conscious experience, personality, and behavior. So we can actually interpret everything on the outside world, process it, and that's how we're going to be using it to make sure that we're a human, make sure we have a personality. We can then affect our behavior based off of what we're seeing and doing. And neurobiology combines the behavioral and life sciences. So neurobiology looks at how neurons in the nervous tissue uses other things than just neurons. And what I mean by saying this is neurobiology studying neurons, but how do the other things that we learned in life sciences affect the neuron? So how do chemicals, how do drugs, how do different pressures, receptors, how are all these different things, these membranes that we looked at earlier in chapter 3, how do all these things used when they come to a neuron? So now we're going to explain what is a neuron. So the endocrine and nervous systems maintain internal coordination. So endocrine is going to use hormones, and endocrine system is going to communicate, but it's going to use these hormones secreted into the blood. The nervous system employs a different system. It uses electrical and chemical means to send messages from cell to cell. A way to kind of think of it is nervous system is extremely quick because it uses chemical and electrical signals. Endocrine's a little bit slow because an endocrine gland has to secrete a hormone. That hormone then has to go into the blood. That blood then has to circulate to the target cell. Also, endocrine system, by using hormones, they have a longer effect than a nervous system. Think of the nervous system as an on and off switch. The endocrine system as more of, you tell somebody to turn the light on, you tell somebody to shut the light off, and whenever they actually turn the light on or shut the light off, that's when they're actually going to be doing the work. So the nervous system has 2 major components: central nervous system, brain, and spinal cord, composed of the skulls of the cranium and the vertebral column. The peripheral nervous system is everything outside of the spinal cord. So it's going to have ganglions, it's going to have nerves, and it's going to have things that travel to glands and muscles. The peripheral nervous system has 2 major functional divisions: sensory or afferent. Afferent means it's coming into the central nervous system. So sensory, you want to have sensory apparatus that comes into the central nervous system, so our central nervous system can evaluate what it's seeing and what it's feeling, and then send that signal out. So sensory is afferent. You have a somatic sensory, which carries signals from receptors, the skin, muscles, bones, and joint, and visceral sensory that carries signals from viscera of the thoracic and abdominal cavities. And every time you hear the word viscera from here on out, it means there is a membrane that surrounds an organ in the thoracic and abdominal cavity. Hearts, lungs, stomach, urinary bladder, they all have these sensory receptors on them. The visceral sensory division carries that back to the brain and the central nervous system. Motor is a little bit easier because motor is efferent, or exiting the central nervous system. So somatic motor is going to carry signals to the skeletal muscles. That's very easy to memorize. Visceral motor, on the other hand, is called the autonomic nervous system. The autonomic nervous system is actually talked about in test 6. It has its own chapter where you take a closer look at the sympathetic and parasympathetic divisions. So the visceral motor is going to carry signals to glands, cardiac muscles, smooth muscles. Involuntary means you have no control over it. You might see the effects of it later on, but you didn't know how you got there unless you're really thinking about it. And these are responses, and responses from the system and its receptors are visceral reflexes because it's from the viscera. So sympathetic, fight or flight. Parasympathetic, rest and digest. Arouse the body for action. Accelerated heartbeat, respiration, while inhibiting digestive and urinary systems. You know that if you've ever been in an area where you had all this rush of endorphins, all this rush of adrenaline, you're ready to fight, you're ready to run. Your pupils kind of change. Your blood pressure changes. Your muscles are ready for action. You know how this feels. So memorizing all these things probably is not important, but kind of think about how you felt. Parasympathetic is kind of like Thanksgiving dinner. Calming effect, slower heart rate and breathing. More blood is going to go to the digestive and urinary systems to digest food, to get rid of waste. That's what the parasympathetic is going to do. And here's a visual orientation of what's in the central. Central is tan, peripheral is yellow. So you can see down the midline, brain, spinal cord. Peripheral is going to be all the outside nerves. We're going to view this later on in the semester, but here's just the central nervous system with the brain and the spinal cord. That's going to be another chapter coming up soon. And the peripheral nervous system, sensory, motor, and then the motor somatic is just skeletal muscle, motor visceral, fight or flight, rest and digest. So now we're just going to look at the neurons. We've gone through all of that background information. And that background information you'll see a couple more times this semester. So if you didn't understand it now, hopefully after the repetition, you'll start to understand how it all fits together. But with neurons, what are the 3 functional properties found in all neurons? That's on the next slide. What are the 3 most basic functional categories of neurons? What are the parts of the neurons? And how do neurons transport materials between the cell body and the tips of their axons? So what are the properties? They have to be excitable. They have to respond to environmental changes, and those changes are called stimuli. They have to be able to conduct and have conductivity. They have to respond to stimuli by producing electrical signals that are quickly passed down to other cells at far locations. Then they have to be able to secrete something. When the electrical signal reaches the end of the fiber, a neurotransmitter has to be secreted that then has an effect on the target cell. So they have to be able to respond to environmental changes. They have to be able to pass that signal to distant areas. Then they have to secrete something that changes a target cell. The functional classes, there are 3 general classes of neurons: sensory, interneuron, and motor. Sensory is bringing signals to the central nervous system. They're usually pain, touch, pressure, baroreceptors, chemoreceptors we talk about later. That does blood pressure and chemicals in the blood. Interneurons are the ones in between the sensory and the motor. The interneurons are actually changing the signal. Maybe it's a very important signal. Maybe if you feel like you're getting really, really, really hot, you might be at the beach having fun. So that interneuron is going to tell the motor neurons to say, no, we're not going to leave yet. We're going to stay here. We're having fun. So we can override sensory neurons by interneurons. And then motor is going to be the action. We're going to move. We're going to do something. The motor neuron can be going to a gland or it can be going to a muscle, but it's still going to produce some type of action. The sensory is going to be afferent because it's going to be detecting stimuli. The interneurons, I kind of mentioned, 90% of all neurons are interneurons because the brain is made up of neurons, and all of those neurons in the brain, a lot of them are interneurons because they are doing some type of processing. The motor, efferent, signals to muscle or glands or the effectors. Here's an example. So the sensory touch, pressure, pain comes into the central nervous system, where an interneuron can say, okay, something's happening here, and then send that off to an efferent or motor neuron that can do some type of action. Now let's look at the parts. Soma, control center of the neuron. This is the cell body. Cell body, in most cells, they're called cell bodies, but in the neuron, it's called the soma. Also called the neurosoma or the cell body. This is where most of the metabolic and regulatory functions occur. There's mitochondria, there's rough ER, there's Golgi, there's all the stuff that you find in a normal cell. They're all going to be found into the soma. It has one central nucleus with a nucleolus, just like a regular cell. And like I said, mitochondria, lysosomes, Golgi, all of these things that are found in a regular cell are all found in a neuron. There's dendrites. Dendrites are usually on the outside, and they're just picking up information from surrounding neurons, from surrounding glands, from surrounding whatever, tissues. Dendrites are the ones that are bringing information to the soma, and the primary site for receiving signals from other neurons. The more dendrites a neuron has, the more information it can receive and incorporate into decision making. It can receive a signal that says, hey, we need to activate ourselves. It can receive another signal that says, no, we don't. So this is how a neuron can start processing information. The axon. It originates from a mound on one side of the soma called the axon hillock. The axon hillock used to be called the axon terminus, is right there. And then at the end of the axon terminus is going to be the axon. You can have an axon collateral that just is going off to the side, or a neuron can just have one axon that goes all the way down to whatever tissue or whatever muscle or wherever it's going. And the axon is specialized for rapid conduction of nerve signals to points remote to the soma or far away. So they have cytoplasm, but here it's called axoplasm. Only one axon per neuron. Sometimes they can collateral or go off to the side. There's always one area that's leaving the soma, and the dendrites usually have many. The Schwann cell is going to be maintaining the myelin sheath in the peripheral nervous system. If you remember in the central nervous system, when we talked about the brain, it was an oligodendrocyte that provides the myelin sheath in the central nervous system. But because these are far away and remote and going through the body, you don't have the ability for one cell to maintain many different axons. Here in the peripheral nervous system, one cell, one node of myelin. One cell, one node of myelin. You can see those little dots right there. All of those are Schwann cells. And one Schwann cell, one node of myelin sheath. And at the distal end, or the end of the axon, there's a synaptic knob. The synaptic knob has all these little enlargements at the end. Sometimes they're called synaptic buttons, synaptic boutons, depending on what teacher you have. They can be terminal buttons or boutons, and pretty much what they do is they form a junction or synapse with the next cell. Each synapse or synaptic knob is going to contain a synaptic vesicle. They're going to have a neurotransmitter. If this neuron was motor and going to a muscle cell, that neurotransmitter is going to be acetylcholine. The same type of thing we talked about when we talked about muscles. They can be multipolar, where they can be multipoles coming in. You can have bipolar, where you can't really tell which one's the axon, which one's the dendrite, because they kind of look the same. You can have unipolar, which means that it's just kind of the dendrites are more developed and longer than the axons leaving out. And you can have anaxonic, where you don't really know what's the dendrite and what's the axon. And these are mostly going to be helping the visual processes. Many dendrites, no axon. There is kind of an axon, but You can't really see what it is because all it does in the visual system is it receives a lot of information and it releases stuff. And it does release stuff onto bipolar cells in the eye. So supportive cells, neuroglia. These are important because if you think about a neuron and how important a neuron is and how important it is in your body, the helper cells, or the neuroglia, are just as important. If you lose the helper cells, you lose the actual glials, you lose the actual neurons. So what are the 6 main types of cells that aid in neurons and state their respective functions? We're going to describe the myelin sheath and why it's important. We're going to describe the relationship of unmyelinated versus myelinated. We're going to look at damage to nerve fibers and how they regrow. So there are around 1 trillion neurons in the nervous system. Neuroglia outnumber the neurons by 50 to 1. So there are 1 trillion neurons. There are 50 trillion neuroglia. Neuroglia, or glial cells, they support and protect the neurons. They bind neurons together, form a framework for the nervous tissue. In the fetus, they guide migrating neurons to their destination. If mature neurons are not in synaptic contact with another neuron, it is covered by glial cells, and these glial cells prevent neurons from touching each other, but also help with the conductive pathway. And they do this because if they kind of form that little synaptic cleft between them, they allow the neurotransmitters to pass and help the conduction down the axon with oligodendrocytes and Schwann cells. So what are the type in the central nervous system? Oligodendrocytes, myelin sheath, central nervous system. They have an arm-like process that wraps around a few nerve fibers and axons that form that insulation layer of myelin. You have epidermal cells. These line the internal cavities of the brain. They're cuboid epithelium with cilia on the surface. They secrete and circulate cerebral spinal fluid. And cerebral spinal fluid bathes the central nervous system in glucose. It bathes it in oxygen. And also it makes the brain buoyant so it doesn't sit down on itself. So epidermal cells, super important. Oligodendrocytes in the myelin, also very important. Microglia, I left that without the red. The reason why is because microglia, there's many classes of microglia, and what they're looking at now in most textbooks and most journals is they're looking at microglia and how they can be used to help in diseases and help in different types of disorders. And because there's so many different types, there's small wandering macrophages formed by white blood cell called monocytes. They kind of complete checkups on brain tissues. They wander in search of debris to just destroy and to phagocytize and to take in. But because there's different types, we're going to leave that kind of out for now. Astrocytes, there's more information on these. They're the most abundant glial cells in the central nervous system. They cover the entire brain surface and most non-synaptic regions of the neurons in the gray matter of the central nervous system. And what they normally do, supportive framework for nervous tissue. They have these little feet that contact blood capillaries. And then this is going to help them keep those blood capillaries and form a tight seal. And astrocytes in the brain form the blood-brain barrier. So if you've ever heard of the blood-brain barrier before, it's the astrocytes that are doing this. Schwann cell we talked about later is in the peripheral nervous system, and it's the one that's going to wind the myelin sheath in the peripheral nervous system. You also have satellite cells surrounded by neurosomas and ganglia in the peripheral nervous system. They provide some electrical insulation around the soma so the electrical processes can pass through, and it regulates the chemical environment of neurons. So satellite cells are also very important. Here's the central nervous system. You can see an astrocyte, and you can see how its feet are going to be surrounding that blood vessel to stop large things from coming through, which is going to create the blood-brain barrier. You can see the oligodendrocyte And because all of these axons and somas are so close to each other in the central nervous system, you can have one oligodendrocyte that can help repair and create myelin on many different types of axons. So what happens if things go bad? So tumors are just masses of rapidly dividing cells. Mature neurons have little or no capacity for mitosis, so they very seldom form tumors. Brain tumors, which you might think that are just growing neurons, they actually come from the meninges, the protective membranes of the central nervous system that you learned about in the brain, the dura mater, the arachnoid matter, and the pia mater. And these metastasize from non-neuronal tumors in other organs. So they kind of come from other places. They kind of go there and they start growing in the meninges. The glial cells that are often mitotically active throughout life can also form brain tumors. So meninges, you can have lung that then will metastasize to the brain. You can have glial cells that can also form brain tumors. You can also have a glioma. These grow rapidly and highly malignant. So blood-brain barrier decreases effectiveness of chemotherapy because chemotherapy drugs are very large, so they can't pass the blood-brain barrier. So with gliomas, you most likely have to have radiation or surgery and then radiation. So now let's look at myelin. Myelin is that insulation layer around the nerve fiber. Think of myelin as that rubber cord around a extension cord, or that cord coming from your cable or from your computer, charging it into the wall. That's what myelin sheath is doing. It's formed by oligodendrocytes in the central nervous system, Schwann cell in the peripheral nervous system, and myelin is 20% protein, but mostly lipids. And if you think about it, lipids are for insulation on the rest of your body, so it makes sense that lipids are going to be used for insulation of axons. And myelination is the production of myelin, begins at week 14 of fetal development. So up until this point, it's not the fact that your axons don't work at week 13 or 14, it's just the fact that the distance between these axons, they don't really need myelin to do anything, because myelin is only going to be used when you're going to be traveling long distances. So during infancy, it produces rapidly, completed by late adolescence. That's why if you see kids run, they kind of run strange. They're still developing those connections between the muscles. They're still developing those parts of the brain, and myelination is occurring all throughout this time. And dietary fat is very important to central nervous system development. And you know the reason why, because most of those fats are going to be made for the myelin that's going to be coating these axons that are extremely important. So peripheral nervous system, we kind of mentioned this Schwann cell. It's kind of just creating different layers. And what it does is it keeps spiraling around. And then every time it lies down, hundreds of layers on its own membrane. And you can see in between them, it's a node and an internode. And that space between them is called the node of Ranvier in the peripheral nervous system. Oligodendrocyte, the same thing. You kind of mentioned this. Multiple nerve fibers, it's going to work. They can't migrate around like Schwann cells do. They're kind of stuck in their own spot. So myelin, many Schwann cells or oligodendrocytes are needed to cover one nerve fiber. Myelin sheath is a segment. I mentioned it. The gap between those segments is called the node of Ranvier. The internode is the myelin-covered segment from one gap to the next. So if you're looking here, you have one node and you have one node of Ranvier. And this is just the oligodendrocyte in the central nervous system. Because there's multiple axons close by, one cell can maintain multiple axons. And this is the Schwann cell in the peripheral nervous system just wrapping around, continually to wrap around, and then that creates one internode. So what happens if something starts to destroy the myelin sheath? Pretty much just as if you started taking scissors and started cutting the rubber sheath around your cable, it would start to leak electricity at those spots. So if you're like, it's leaking electricity, it's not carrying all the same electricity to your computer or to your TV or whatever you have. The same thing happens in the body. So if we're looking at myelin sheath and it's being damaged in different parts, one of them is multiple sclerosis. Oligodendrocytes in the myelin sheath in the central nervous system, they start to deteriorate. Myelin, instead of being replaced over and over again, is now going to be replaced by scar tissue. That's hard. It doesn't conduct the system. It doesn't have as much lipids in it. What happens is it doesn't allow the signal to pass through it. So sometimes the nerve impulses stop. Sometimes they skip. Sometimes they get damaged. And what happens? Double vision, tremors, numbness, speech defects. The problem with multiple sclerosis, onset between 20 and 30, but right when you get it, 25 to 30 years later after diagnosis, you'll probably die or be dead. It may be caused by autoimmune deficiency triggered by a virus when you're younger. They don't really know what triggers it yet. But what happens if you think about this, if you have multiple sclerosis, you start having this scarring on your axons. Soon, the nerve impulses going to your respiratory system will stop working. So it means you can't breathe on your own. You can't breathe on your own. That's why 25 to 30 years after your first diagnosis, you're usually dead, because it starts to progress to parts of the brain and parts of the systems in your body that you can't overcome. You become shaky and all this other stuff. So Tay-Sachs. Tay-Sachs is also a myelin sheath deficiency. It's a hereditary disorder of infants from Eastern European Jewish ancestry. And it's the abnormal accumulation of a certain type of lipid called glycolipid GM2. What happens when this starts to build up in the myelin sheath? Normally, this glycolipid gets decomposed by a certain enzyme. If you have Tay-Sachs, you're missing this enzyme. So accumulation of GM2 disrupts the conduction of the nerve signals. Blindness, loss of coordination, dementia, fatal before age 4. And the reason why Tay-Sachs is so bad and kills you before age 4 is because when you are building all of the myelination, when you're a fetus, when you're 1, you're 2, you're 3, you keep building up this glycolipid. This glycolipid disrupts the conduction of the nerve, and usually by the age of 4, you have so much of this glycolipid built up that the nerves cannot send signals to where it wants to go. If you have unmyelinated nerve fibers, you can kind of sit there and think, you're like, okay, why would you have an unmyelinated nerve fiber? An unmyelinated nerve fiber maybe is just going a short distance. Maybe that unmyelinated nerve fiber wants to be touching some tissue or some fluid, so it's more sensitive. So touch. You can have places in the brain, the spinal cord that has unmyelinated nerve fibers because they're only going very short distances. The interneurons are very close by. So a Schwann cell, 1 to 12 small nerve fibers in grooves on the surface, membrane folds once around, keeps wrapping around, and you can see this is myelinated versus an unmyelinated nerve fiber right there, unmyelinated. So I kind of mentioned this. Unmyelinated would slow down the conduction of the fiber. Maybe it coming a little bit slower to a target cell is important. Maybe it's for processing of something else. But what we're going to be looking at here, how can we create the fastest speed of a nerve fiber? To do that, you want to have a very large diameter. The larger diameter, the more surface area around the axon. The more surface area around the axon, the quicker it'll go. If it has myelin around a very large diameter axon, it moves even faster. So large fibers that have more surface area and conduct signals more rapidly, and then myelin's going to make it the quickest. So if you have a small unmyelinated nerve fibers, they kind of move at around either 1/2 to 2 meters per second. That's still pretty quick. You can have a small myelinated fiber that speeds it up to 2 to 15 meters per second. If it has a large diameter and it's myelinated, up to 120 meters per second. And if you think about that, your largest neuron, the axon actually, is about 1 meter long. If you're around 5 feet tall or 6 feet tall, it can be a little bit taller or a little bit longer. But think about one going through your brain all the way to your toes. If you wanted to wiggle your toes, how long does that take? Doesn't take even a second. It's a fraction of a second because that axon that's going to your toes is large diameter. It has a coating of myelin on it, so it's moving at up to 120 meters per second. Slow signals supply the stomach and dilate the pupil. Speed is not really the issue here. The fast signals with the large myelinated, like I said, skeletal and transport sensory signals from vision and balance. Vision and balance is very important. If you're feeling wobbly, you don't want to fall over. And that completes this micro lecture.